One ATP sacrifices one phosphate group to add to the glucose. The glucose is then rearranged to form fructose. One more ATP sacrifices a phosphate group. The fructose is then split into two 3-carbon sugars which are isomers of each other. Only one of these sugars can be processed in glycolysis, so the one that cannot be processed is changed into the one that can. Each of these molecules sacrifices a hydrogen ion and has a phosphate added to it from the cytosol. The phosphate is then broken off of each to make 2 ATP. These new molecules are then rearranged, and 1 water molecule is taken from each. Finally, the remaining phosphates are made into ATP, and we now have 2 molecules of pyruvate. Each molecule of pyruvate then loses 1 carbon dioxide, 1 hydrogen ion to NAD+ to make NADH, and has coenzyme A attached to it. Coenzyme A is stripped from acetyl-CoA. The resulting molecule then has 1 water molecule removed and another added. This then sacrifices 1 carbon dioxide and 1 hydrogen ion to NAD+ to make NADH. This molecule then loses 1 more carbon dioxide and 1 more hydrogen ion to NAD+. This then has coenzyme A attached, which is replaced by a phosphate group, which is then taken away to make another ATP. FAD then comes and takes 2 hydrogen ions. 1 more water molecule is added, and 1 more hydrogen ion is sacrificed to NAD+. Pretend for a moment that the gray pieces that normally represent hydrogen are now electrons. This is the Electron Transport Chain, where electrons are shuttled between membrane proteins. This next path is the one that hydrogen ions would take in the Electron Transport Chain. Chemiosmosis is where hydrogen ions spin ATP synthase to combine phosphates from the cytosol and ADP to create ATP. 32 to 34 ATP are created, meaning that it is not a 1-to-1 ratio. In this step, 2 pyruvate each lose 1 carbon dioxide to make acetaldehyde. This is then given 2 hydrogen ions to create ethanol. In lactic acid fermentation, 2 pyruvate are directly reduced by NADH to lactate,